Fingerprinting. As usual, I'm your chemistry teacher, Mr. A.Dimitri. So the first step is to collect a material which contains DNA. So for example, blood, saliva, hair follicles, bone, etc. Then the next step, after getting this material which contains DNA, the next step is extraction. So extraction is, so we want to separate DNA from the protein in the chromosomes. So this process involves in breaking up the cells so that you can release the DNA. Then we separate the DNA from the protein and the cellular debris. Then also involve precipitation of the DNA with alcohol, then cleaning the DNA. Then after that, you can either confirm that the DNA is clean or not. Then after extracting the DNA, the next stage is cutting DNA fragments into smaller pieces using restriction enzymes. So we have a variety of restriction enzymes. Each cut DNA at specific points. Then these specific points, they contain mini satellites. Then these mini satellites, they are unique, as similar to our fingerprints. Then so sometimes the DNA quantity is very small, so we have to amplify the quantity of DNA we have. So we transfer the DNA into another medium which contains some free bases. So in that setup, we are going to have our enzyme which can amplify the DNA, which is DNA polymerase. So it will undergo a chain reaction, a polymerase chain reaction, so that we can amplify. Because if we use a smaller quantity of DNA, so the resolution becomes very low, so it becomes difficult to analyze that DNA. So the process of polymerase chain reaction, it became popular during the COVID era when we were testing for COVID. So like in this case, the process of amplifying the DNA, for example, let's say we have got the virus we are using the COVID virus. So the first stage, so at room temperature at 20 degrees Celsius, the DNA has got its helical structure due to hydrogen bonding. So the first stage is denaturation, so that we can break the hydrogen bonding. Then next stage is primer annealing, so that we are adding a primer at the end of the sequence of the DNA strand. Then the DNA polymerization using polymerase will occur at temperature of 70 to 74 degrees Celsius. Then after that, so we are using free bases in the medium. The next stage is denaturation again. We separate the strands, then we repeat primer annealing, DNA polymerization, then we separate the process, keep on repeating. So the quantity of the concentration of the DNA is increasing. So let's say we now have our DNA, then back to normal temperature, the helical structure is restored. Then since we now have this quantity of DNA strands, large quantity of the DNA short fragments, then the next stage we are going to carry out electrophoresis. So electrophoresis separates the DNA fragments based on their MR. So DNA fragments, they are negatively charged because of the phosphate groups. So we are going to place them at the cathode, so they are going to be attracted towards the anode. So the short DNA fragments will travel a longer distance than the longer DNA fragments. So in the next stage, so like in this case, so we have collected the DNA, then we use the restriction enzymes as well as the polymerase reaction. So we are going to start the electrophoresis by starting our machine. So the bases, they are moving towards the positively charged anode because our DNA bases, they are negatively charged. So the process is carried out in agarose gel, which is a sugar which is extracted from seaweed. So this is gel electrophoresis. So as the particles, the bases are moving, so these are shorter. So this base contains 50 base units, while this one has got 76. So the shorter DNA fragments, they travel a longer distance. Okay, so after carrying out electrophoresis, so we want to analyze our DNA. So how do we do that? So depending on the setup, we can use nylon membrane to to extract these bases onto a nylon membrane. Then we place the nylon membrane containing DNA fragments in a medium which contains phosphorus 32. So remember, we have got phosphorus 32 isotope and phosphorus 31, but the phosphorus 32 isotope is radioactive, so it emits radiation. So it becomes easier so that we can record the DNA, the phosphorus 32 isotope will stick to the DNA fragments, then will start to emit radiation. So on X-ray film, the the spots where DNA fragments were present will be lighter, while where the points where we do not have the DNA fragments or the absence of phosphorus 32 will appear dark. So in this case, I'm going to use UV light, so that I'm going to use a fluorescence treatment. So in the presence of fluorescence, in presence of fluorescent light, the DNA bases will appear, will start to glow. Then you can shoot a film in the presence of that light. So here, for example, this part contains 50 base pairs, then this one 100 base. Then so the heavier the fragment, the shorter the distance it travel. So the intensity of the bend will depends on the quantity of the base pair of the fragments on that size. So after getting this, we can compare. Like in this case, we're having two. We're comparing, for example, let's say a victim as well as the other one for a crime scene. Okay, so after we get the DNA fingerprint. So after that, we can use that one. It's either for victim for a crime scene. We expect 100%, though there is mixing of evidence of DNA material, because a crime can be committed by more than one person. Okay, so in the next slide, I'm going to show you some DNA fingerprints of a crime scene as well as of suspects, and then you can choose which one committed the crime. I thank you.